Not the last day. Yes. Hallelujah, right? I feel the same way you guys do. It's a long haul. So we have one batch of material to get through. I did pass back, as I noted on the email, your final exam. Or not final exam, your second exam. Final exam is tomorrow, which I'll be writing tonight. And key is posted outside my office, and I will be around if you have questions. I'm also going to make myself available for questions after class again today, as I did before, and I will videotape that, and so get things posted as quickly as I can tonight. That's what I can assure you, and believe me, I want to get it up as quickly as you guys want it up. Maybe not quite as quick as you want it up, but I'll get it as quickly up as I can, because it does take a while to do the renderings. But I can assure you that I'll work on that very hard tonight. Let's see. So we are at the point of talking about glycogen metabolism. And glycogen metabolism is a really good place to end the term, because a lot of the things that we have talked about to this point come together. We see, you started to see this in the last lecture, though it was complicated. You started to see how the hormones that we learned about before were interplaying and controlling the processes of either glycolysis or gluconeogenesis. You're going to see more of that today. You're going to see glycogen being controlled by hormones, whether for synthesis or for breakdown. And what you will see today is a very good demonstration of reciprocal regulation. You saw it yesterday in an allosteric sense, and also in a sense of synthesis of F26BP or its breakdown. Yes, sir? Epinephrine and glucagon are hormones. Yeah. And I'll also talk about glucagon a little bit today as well. Okay? So, so what you'll see is, like I said, that these things really all do come together. There is some complexity today, and that complexity will come mostly near the end again. So we probably will finish a little bit early today, because as you will see in the first part of what I have to say today, glycogen metabolic pathways are extraordinarily simple. There's very, very little to them. There's about 2 enzymes, 3 enzymes to consider in each of them, and that's basically it. And there's only one enzyme in each pathway, the synthesis and the breakdown pathway, that is regulated. And they're reciprocally regulated. So in a sense, it's actually a relatively simple metabolic pathway. As you will see, the regulation of these is a bit complicated. So I'm going to go through it kind of slow, probably a little slower than I've gone through the other lectures, just because I've got a little bit of time to do that. Well, let's go back and refresh our memories with respect to glycogen structure. Glycogen structure, you recall, glycogen is what I described earlier in the class as an animal polysaccharide, and it is a polymer consisting solely of units of glucose. Solely of units of glucose. Most of the links between the glucose molecules in glycogen are alpha-1,4 linkages. But wherever there's a branch, as you see here, there is an alpha-1,6 linkage. So we have an alpha-1,6 linkage, but then everything after that is 1,4 until the next branch, etc. Glycogen differs from the plant. Boy, I have the hiccups today. Excuse me. Differs from the plant polysaccharide known as amylopectin, in that glycogen has much more branching. Glycogen branches on average about every 10 residues. Amylopectin branches on average about every 30 to 50. And I told you I would tell you why that's very, very important for animals. So we're at about that point of talking about that. And if I take a look at the structure of glycogen schematically, what we discover is that it looks something like this. Okay? So here's a schematic representation of glycogen. Here is our very first glucose that's here. We go a few residues, we see a 1,6 branch. That 1,6 goes 1,4 for a ways, then it has a 1,6 branch, etc. And so each of these branches themselves can have branches and branches. So the more frequently we have branches, the larger number of ends of the molecule that we will have. Now that's the key to glycogen's structure from the perspective of an animal. All right? Large numbers of ends are important because, as you will see when I talk about the breakdown of glycogen, that the breakdown of glycogen occurs from the ends. The more ends that there are, the more glucoses that can be released at once. So glycogen breakdown occurs from the ends. More ends translates to more glucose. And of course, for an animal, that's very important. Animals have to move fast. They have to escape predators. They have to catch prey. They have to do a variety of things. And a lot of glucose very quickly facilitates that. Plants don't need to move fast. Plants don't have rapidly changing energy needs. And consequently, the number of ends that they have isn't as critical as it is for an animal. So the structure of glycogen really facilitates life as an animal because it can release a lot of glucose very quickly. What we will see as I go through talking about glycogen breakdown, and I will spend more time talking about breakdown than I will synthesis. As I talk about breakdown, what you will see is that the body, or the cells of the body, actually have two different ways of activating glycogen breakdown. It makes it a bit more complicated, I'll be honest with you. But two ways allows for an instantaneous response and a rapid response. Okay? So instantaneously happens as the needs of the cell drastically change from second to second. A rapid response is the hormone response that you've seen before. Well, what's an instantaneous response? I'll give you a clue. It's allosteric. Allosteric responses happen almost instantaneously. Glycogen has built into its regulation both of those responses. Okay? Now that's a very, very important thing because, as we shall see, the needs of the body, the needs of the cells in the body change drastically. Okay. Well, we start our discussion of glycogen breakdown by talking about the first enzyme in the pathway. The enzyme that is the most important enzyme in glycogen breakdown is known as glycogenphosphorylase. Glycogenphosphorylase catalyzes the reaction that you see on the screen. And it's a different kind of a reaction than we've talked about before. Okay? A phosphorylase we haven't talked about. We've talked about a phosphatase, but a phosphatase is doing something very different from what a phosphorylase is doing. Okay? So we need to consider for a moment what this enzyme is catalyzing. As you can see in the figure, glycogen is basically having one of those glucoses clipped off of the end. So glycogen phosphorylase is breaking an alpha-1,4 bond. Okay, glycogen phosphorylase only works on alpha-1,4 bonds. And here's the hook. It doesn't use water as a cleaving agent. It uses phosphate. And that's why we call it a phosphorylase. Otherwise, it would be known as glycogen hydrolase. And you might think, well, big deal, so what? Phosphate versus water, is it? Who really cares? Well, there is one consideration for that, and the consideration for that is that look at the products that we get. Here's glycogen on the right with one less residue than we had before, and here's glucose 1-phosphate. Okay? Now, glucose 1-phosphate can readily be converted into glucose 6-phosphate. From an energetic perspective, they're essentially equal. Now, that's an important consideration because when we think about how did we make glucose 6-phosphate from glucose in the cell, what did we have to have? ATP. Hexokinase require glucose plus ATP to make glucose 6-phosphate. Well, here is a reaction in which we are making the equivalent of glucose 6-phosphate without using any ATP. How does the cell do that? What magic is going on here? Did we just defeat the energy barriers that are there? Well, the answer is we never defeat energy barriers. The energy always wins out. So there's a reason why this reaction can proceed without ATP. What is the reason? Last day of class, nobody has any thoughts left in their head, right? It's not a hard one. The reason is because that there's energy in an alpha-1,4 bond. The breakage of that alpha-1,4 bond releases energy, and that energy is used to put the phosphate on. So we don't need ATP if the cell is efficient at harvesting that energy, and of course it is in the form of glycogen phosphorylase. So the cell has just saved itself 1 molecule of ATP for the first glucose that it has broken, first glucose 1-phosphate it has broken off. It's like a get-out-of-jail-free card. Okay? It is going in this glucose 1-phosphate, as we shall see, gets converted to glucose 6-phosphate, and bang, it's in glycolysis without any ATP investment whatsoever. Okay? So that's kind of a cool phenomenon. So this reaction is called a phosphorylasis. Okay? P-H-O-S, P-H-O-R-O-L-Y-S-I-S. A phosphorylasis uses a phosphate to break a bond. So a phosphorylase catalyzes a phosphorylasis. Okay, well, so we've reduced the size of glycogen by 1 residue. We've made a glucose 1-phosphate. Okay, let's look. I'm going to jump down here and let's look to see what happens with that glucose 1-phosphate. Glucose 1-phosphate is converted by an enzyme known as phosphoglucomutase to glucose 1-phosphate. You notice the reaction on the screen. First of all, there are no ATPs involved. So again, all we're doing is just rearranging the phosphate on that molecule. And you'll notice that this guy had as part of its name, mutase. And I told you that a mutase had unusual mechanisms of catalysis. It was the phosphoglycerate mutase, for example, that produced the 2,3-BPG as an intermediate that it released. Okay? This mutase is doing something very similar. Here's glucose 1-phosphate, but rather than just move the phosphate from here over to here, the mutase starts by putting a phosphate on one side and taking it off of the other side. The result of that is that we have glucose 6-phosphate, and now glucose 6-phosphate is bang, it's in glycolysis. So just that quickly, if this reaction happened in a muscle cell, which it could very easily, if this reaction happened in a muscle cell, the muscle already has an energy pathway going. 1 step, 2 steps, bang. Phosphorylasis, mutase, and it's ready to go for glycolysis. So it's very rapid. It gives that cell that rapid burst of energy that it's going to need. Okay, well, there's only one other enzyme we have to take into consideration in glycogen breakdown. And the clue was that the glycogen phosphorylase only worked on alpha-1,4 bonds. All right? Well, that means we've got to have something for dealing with those alpha-1,6 bonds, and those are dealt with by an interesting and odd enzyme called debranching enzyme. It has a big mouthful of a name, and we're going to call it debranching enzyme. Debranching enzyme actually has within its protein 2 catalytic activities. 2 catalytic activities. We'll see how they are here. What you see on the screen is a branched glycogen molecule. You see the 1,6 branch. And what you see above on the top thing is commonly what debranching enzyme would see. The reason it would see something like this is glycogen phosphorylase not only will not cleave a 1,6 bond, it won't even cleave close to 1. So it will get to within 4 or 5 residues. They show 4 here, and I don't count residues, so I'm not overly concerned with that. But it gets within a certain distance of a branch, and glycogen phosphorylase stops. That leaves this template for debranching enzyme to fix, basically. And it takes 2 catalytic activities for debranching enzyme to do that. Okay? First, you can see that it I'm sorry. So the phosphorylase doesn't go to here. I'm saying the wrong thing. Phosphorylase goes to here. So it cleaves off the red. All right. Phosphorylase works to this point. It's stuck here. It doesn't go any further than where I have my arrow right here. All right. It gets to that point, debranching enzyme comes in. It's what they're calling transferase. So the two activities in debranching enzyme have these named, transferase and this. We call both of them debranching enzyme. All right? So both of these are activities of the debranching enzyme. Debranching enzyme grabs 3 of those guys and moves it and makes 1,4 bonds. It moves those 3 out here, makes 1,4 bonds, and so now we have a longer chain of alpha 1,4s out here, and we're left behind with a single alpha 1,6. At this point, the other activity of the debranching enzyme uses water to cleave off that last glucose. Now, based on what I just told you, you probably should find that a little surprising. Why does it use water and not use phosphate to clip that glucose off? I can outwit you guys this time. Why does it use water and not phosphate? Why did it use phosphate in the first place?
There less energy in the alpha 1,6 bond?
Exactly. There is less energy. So you guys, you can do it. That's good. There's less energy in a 1,6 bond than there is in a 1,4 bond. That's why, for example, the enzyme doesn't grab this 1,6 and turn it into a 1,4, because there's not enough energy in a 1,6 to convert it to a 1,4. There is enough energy in a 1,4 to convert it to a 1,4, which is what it's doing, but not a 1,6. So it leaves the 1,6 behind, comes back later with a water, clips it off, and as a consequence now we have a molecule that is less branched than it was before. Now glycogen phosphorylase can come along here, start clipping off end, until it gets to another place where there's a branch. Okay. That's all there is to glycogen breakdown. I told you it was a simple pathway. That's really all there is to glycogen breakdown. Yes, ma'am? I'm sorry? Core is the core of that is the rest of the glycogen molecule. So we saw that big branched structure before, that's the core. We're not going to play this game today. Okay. Go back here. All right. So let me get my pointer, and then I'll just deal with the pointer. That way I won't go through all the crap of that thing. I thought I had that issue fixed, but obviously I don't. Okay. So any questions on this? Make sense? Okay. It's not very complicated. Glycogen phosphorylase is a fairly straightforward phenomenon. Okay. Glycogen phosphorylase has some interesting considerations with its structure, but I don't think for our purposes that they're probably essential, very essential for us to worry about. What's more important for us is to think about the enzyme and its regulation. And the enzyme's regulation is yet another enzyme that is regulated by both covalent means and by allosteric means. You saw pyruvate kinase, for example, was regulated by phosphorylation, dephosphorylation, and also by allosteric effectors. The same thing happens with the glycogen phosphorylase. Now, glycogen phosphorylase exists in 4 different forms. So I'm going to spend some time going through these forms, talking about them, and then move forward. So what are the 4 forms of glycogen phosphorylase? Well, there are 2 forms that differ from each other in the presence or absence of phosphate. Okay? They're known as the phosphorylase B. This is, whenever, by the way, most people call this phosphorylase. I kind of prefer myself to call it glycogen phosphorylase, but even I slip and call it phosphorylase sometimes. Okay? Most people just call the damn thing phosphorylase. But it's glycogen phosphorylase B. The B form has no phosphate. It is the B form that most people think of as being the less active form. Now, as we will see, the B form can be active. But most people think of the B form as being less active, and you'll see later why people think of it as being less active. Glycogen phosphorylase A, you'll notice, has two phosphates. By the way, this is a dimer of two identical subunits. Each one gains a phosphate, and that converts the enzyme from the B form into the A form. Okay? As you might imagine, by the addition of phosphates, there are structural changes that happen in the enzyme, and those structural changes in the enzyme change the way it behaves, basically. And the way it behaves is with respect to allosteric effectors. Okay? Now, in addition to the B and the A forms of the enzyme, each of the B and the A forms can exist in an R state, which is more active, and a T state, which is less active. So we can have glycogen phosphorylase B in the T state. We can have glycogen phosphorylase B in the R state. We can have glycogen phosphorylase A in the T state, and we can have glycogen phosphorylase A in the R state. So all four forms are found. Now, the conversion from R to T and T to R, these are allosteric conversions. They're always allosteric. R and T conversions are always allosteric. So we will see that there are allosteric effectors that change R to T in the B form, and there are different ones that change phosphorylase A in the R to T forms. Now, in addition to that, the phosphorylation is hormonally controlled. So putting the phosphate on, taking the phosphate off, depends on hormonal pathways you've already seen. I'll refresh your memory on those in just a minute. Okay? Okay, so that's what happens with these guys. Let's take a look now at their regulation. Your book makes a kind of a production of these things being in the muscle versus being in the liver. And while they are important considerations, I don't want muscle liver to confuse you. If you get confused about things, then go back and think about what muscles are doing compared to what liver is doing. But I'm not going to ask you specifically different things about muscle and liver. All right? Well, the glycogen phosphorylase that we're examining here is a glycogen phosphorylase B that is found in muscle. Okay? So we're looking only at the B form. We are understanding how it is controlled. Well, when we look at this, we see that we go towards the leftwards direction. We are going into the T state. The T state is the less active form. As we go to the right, we're going into the R state. The R state is the more active form. Okay? Well, look at the allosteric effectors. The two allosteric effectors that take it to the left, that is in the T state, are ATP and glucose 6-phosphate. What does ATP tell us about the energy of the cell? It's high. When ATP levels are high, the cell has plenty of energy. When the cell has plenty of energy, does it want to be breaking down its glycogen? No, it doesn't, any more than it wants to be lighting its fireplace on a 90-degree day. All right? So high energy states, and yes, glucose 6-phosphate will also indicate that. All right? High energy states are favoring the inactivation of glycogen phosphorylase B. All right? Now, let's imagine that I am a muscle cell and I'm out doing my contraction, let go, contraction, let go, contraction, let go, etc. And I'm doing a lot of that. What's happening to my ATP levels? Well, my ATP levels will start falling almost instantaneously. And if they don't get relief, ADP will start to accumulate, and as ADP starts to accumulate, it will slowly get converted into AMP to provide energy to cells. AMP, therefore, is an indicator of low energy. So when cells have low energy, glycogen phosphorylase B in the muscle gets converted into the R state, and now glycogen phosphorylase B starts breaking down glycogen effectively. This is a backup. This is a backup. Why is it a backup? Well, let's imagine that the hormone hasn't arrived to phosphorylate glycogen phosphorylase B. I want to have another way of turning this enzyme on if I'm in a desperate situation, and AMP is definitely telling me I'm in a desperate situation. Hell, we're not going to wait for the hormone. We're going to start breaking down glycogen because we need glucose because we've got to contract. All right, so we see that glycogen phosphorylase B can exist in a very low energy, that is a low catalytic state, the T state, and a high catalytic state, the R state. So why do we think of glycogen phosphorylase B as being the less active form? Because most of the time it will be in the T state. Most of the time the cell has plenty of ATP. It has plenty of G6P. And not very often does it ever have much AMP. So if we looked at all the glycogen phosphorylase Bs in all of our cells, we would discover that for the most part, it's in the T state because there's rarely that much AMP floating around to activate the enzyme. So it's for that reason people think of it as the less active form. Does that make sense? Okay, let's take a look at glycogen phosphorylase A. It's actually even a little bit simpler. Glycogen phosphorylase A, this one is studied in liver, can exist also in two states. It can exist in the T state, and it can exist in the R state. And again, the conversion between those two is allosteric. As we move to the right, we are favoring the R state. As we're moving to the left, we're favoring the T state. Okay. Well, what is happening with these two guys here? All right, glucose, that is free glucose, converts the enzyme into the T state. When is a liver cell going to have plenty of glucose? Well, let's think what the liver is doing. Here's the liver cell. It gets a message that we've got danger. The grizzly bear is chasing me. Okay. Epinephrine binds, we start breaking down our glycogen, we start synthesizing glucose and gluconeogenesis. Those two together start producing glucose, and the liver starts dumping glucose out into the bloodstream. We're starting essentially the Cori cycle. Glucose travels to the muscle tissue which uses it. And as long as that process continues, the blood concentration of glucose stays low because the muscles keep taking it up. Now, when I get away from the grizzly bear and I'm stopping and I'm panting and I'm catching my breath, the muscle cells are no longer contracting. They get caught up with whatever glucose it was that they wanted, and they finally go, okay, I got plenty of glucose. What happens to blood glucose levels? They start going up because the muscles aren't taking them anymore. As the blood glucose levels go up, what happens to the liver glucose levels? It too starts going up because there's no place to put it. Glucose is a poison. Got to do something with it. We certainly don't want to be making any more glucose 1-phosphate from glycogen, and therefore the enzyme flips into the T state and says, hold on, quit making this stuff. Okay? Now, how do we get to the other direction? How do we go from T to R? T to R is even simpler. When there's no glucose, the enzyme's in the R state. So as long as the cell isn't full of glucose, glycogen phosphorylase A is in the R state, ready to break down glycogen. It's for this reason we think of glycogen phosphorylase A as being more active. Okay? Much more commonly is it going to be in this state than it's going to be in this state, because much more commonly is there not going to be any glucose around than there is going to be much glucose around. Okay, makes sense? Questions? Giovanna?
So, this is going back to the other thing I was going to ask.
Yep.
We're talking about the debranching.
Mm-hmm.
This is one of those, do we need to know it? Can you just talk about those two enzymes that are there as the debranching, or are we supposed to break it out?
No, they're both, the breaking off of the branch and the 1,6 bond breaking, both of those are part of debranching. So no, I'm calling them both the same thing. They're both called debranching enzyme. Yeah. Okay. Other questions? That is glycogen phosphorylase A. All right. Now, let's see. Oh, I think I jumped ahead there. Hold on. Here's what I want right here. Okay, no. Here's what I want right here. No. Here's what I want right here. There's what I'm after. Okay, I'll come back to the other figure in just a minute. All right. Now, this figure is to remind you of hormonal control. So now you've seen the complete activation of a pathway relative to hormonal control. Grizzly bear stimulates production of epinephrine. That's adrenaline. The other hormone that can be released is called glucagon. Okay? Glucagon mimics what happens with epinephrine. It mimics what happens with epinephrine. Glucagon only works on the liver. It only works on the liver. Epinephrine will work on muscle or liver, but glucagon will only work on the liver. I like to think of glucagon as telling us glucose all gone. Because when blood glucose levels fall, even if you're not being chased by a grizzly bear, your blood glucose levels can fall, right? Glucagon is released to stimulate the liver to make some glucose and keep those levels from falling too low. Because if your glucose levels fall too low, you're hypoglycemic and you're in trouble because your brain, in particular, is dependent upon glucose. It can't make its own, and it's sending a signal to the liver saying, hey, kick up the blood glucose levels or I'm going to pass out. So glucagon stimulates essentially the same pathway that epinephrine does, the difference being it only stimulates it in the liver. Now, you've seen what happens. Hormone binds to a 7-TM. 7-TM activates a G protein. G protein drops its GDP, picks up GTP. That causes it to release the beta and gamma subunits. You've seen all this before. Okay? The activated G protein interacts with adenylate cyclase. Adenylate cyclase, in turn, catalyzes the formation of cyclic AMP. Cyclic AMP activates protein kinase. And now we're getting ready for the end part of the story. The end part of the story is this. Protein kinase, as its name suggests, phosphorylates proteins. You saw how it phosphorylated pyruvate kinase the other day. Now you see how it phosphorylates phosphorylase kinase. Phosphorylase kinase is an enzyme. It's, again, probably better called glycogenphosphorylase kinase, but if you call it phosphorylase kinase, that's fine as far as I'm concerned. Phosphorylase kinase in the dephosphorylated state on the right in red is inactive. When it gets phosphorylated, it gets converted into an active state. Now, you may remember when I talked about blood clotting that I said cascades allow for amplification of the signal at every step. This may make a few cyclic AMPs, which may activate a few protein kinases, but it's going to, each one of those protein kinases is going to make many phosphorylase kinases active, and those many phosphorylase kinases are going to make many more glycogen phosphorylases active. Okay? So this cascade that's happening from here to here to here to here is allowing the cell to make a ton of glycogen phosphorylase A, and to do it very, very quickly. Now again, animals have these quickly changing energy needs. This serves those needs very, very well because the more glycogen phosphorylase molecules we have that are active, the more ends of those glycogen molecules can be clipped to yield glucose 1-phosphate. Okay, so this is a tidy system. At this point, we've shown now how we've gone from having glycogen phosphorylase with no phosphate on it to glycogen phosphorylase with a phosphate on it. Don't forget this enzyme, phosphorylase kinase. Okay? It's not protein kinase A that converts this. It's phosphorylase kinase that makes the conversion between glycogen phosphorylase B and glycogen phosphorylase A. Okay. Now, that's glucagon, if anyone wants to memorize the sequence of amino acids in glucagon. And now I want to show you a little bit about how the complicated, or I shouldn't say complicated, but the more complex scheme is for controlling phosphorylase kinase. On this scheme here, it looked like, well, phosphorylase kinase, it simply has to get a phosphate put onto it and it's active, right? Well, that's fine and dandy if the only way we activate that enzyme is when a hormone tells us to. Let's imagine the following situation. Let's imagine that I am sitting here and this room catches on fire. Okay? My adrenaline will get flowing. I have to get up and I want to get out of here as quickly as I can. In the matter of the first few steps, I can burn up a pretty good chunk of the ATP that I had stored in my muscle cells, in my legs. Fairly quickly, my glucose levels fall. You saw how creatine kinase kicks in. It's going to make up some of that difference. But I'm waiting for epinephrine to move from my adrenal glands to my liver to my muscle cells. Okay? That takes a few seconds. In the meantime, I need to get out of here now. I really would like to have a second mechanism of activating phosphorylase kinase quickly that isn't relying on a hormone. Okay? Well, it turns out that phosphorylase kinase has built into it two regulatory systems. They're actually shown right here. It's not as complicated as it looks. What's that? Yeah, sure, right? Okay, I'll slow down. So, the aim here is we want to have phosphorylase kinase have two regulatory systems. Over here, completely inactive phosphorylase kinase. All right? No fire. I'm sitting and relaxing, watching everything go on. No panic, no nothing. Okay, my phosphorylase kinase, I really don't want it active because they don't want my glycogen breaking down when I'm not doing anything. Okay? Fire alarm goes off. I got to get out of here. I got to run. I got to get going before the epinephrine hits my system. What's the first thing that happens when my muscles start contracting? Well, we didn't talk about it in this class, but I'm sure you've talked about it in physiology class. The signal for muscle contraction are calcium ions. Calcium is released, and the calcium release in muscle cells stimulates contraction. So muscles that are contracting will have increased concentrations of calcium in them. Calcium alone can partly activate this enzyme. So from the very first instant that muscular contraction happens, some of the phosphorylase kinase becomes activated instantaneously. It doesn't happen much faster than that. Okay? Now, how do I get the guy completely active? Well, here it's partly active. To get it completely active, I've got to not only get calcium, I've also got to get the guy phosphorylated with calcium. That gets me over to here. So we could think of the first step with calcium as being partly active. Only when it gets phosphorylated with calcium is it fully active. And that's what this is trying to show you. Is it possible for it to get phosphorylated without calcium? Yes, it is, and that's what's happening on the top. What if the hormone binds and the muscles haven't started contracting? Well, that's fine. Okay? We can, in fact, phosphorylate it. When calcium comes along, it's going to make it fully active. Both of these together make the enzyme completely active, and when it's completely active, you can bet your bottom dollar that a lot of phosphorylase B is getting converted into phosphorylase A by this enzyme. This enzyme catalyzes that conversion. Questions about that? Yes?
So the upper method that you're talking about is what would happen if it was strictly relying on hormones?
That's correct.
And then with calcium, that's what makes it go quicker?
That's correct.
Okay.
Yes, sir?
Are those two that you were talking about, instantaneous and rapid?
So the instantaneous part is the calcium.
And the rapid
The hormone is rapid. That's right. So hormone is rapid because it flows through your bloodstream, but it's not instantaneous. This guy is instantaneous. As soon as your muscles start contracting, that calcium's there. Yes, sir?
So it takes the bottom and the upper to make it fully active?
That's correct. But either one of these alone, either just the calcium or just the phosphorylated, will start doing this conversion. Yes, ma'am?
Sorry, what exactly is going on with like the center lines? Like those are kind of confusing for me.
Okay,
Like, where
so
it
it's just
jumps.
simply, it's a dumb figure. It's just,
Okay.
it's trying to show you that once you get calcium on it, you got to put a phosphate on it to get here.
Oh,
Or if you
okay.
put a phosphate on it first, you got to get some calcium on it to go here. Yeah,
Okay, all
it's
right.
kind of dumb. I think they could draw the figure a little better. Okay. Well, we may be actually at a stopping point. Let me see. I think that's a stopping point. It is a stopping point. Okay. Let's take a brief break, and we will finish. We actually are going to finish early today, and come back and talk about glycogen synthesis and its control.